This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 67, recorded on February 4th, 2014. Hi, everybody. I'm Vincent Racaniello, and with me is Dickson Donald Despommier.
Oh, that's a very formal introduction, Vincent.
Did I get the middle name right? It's Donald,
you did.
Right?
You did. There's an interesting story that goes with that, but I don't think this is neither the time nor the place.
Donald Trump?
No, it has nothing to do with Donald Trump nor Donald Duck. It's got something to do with my past, but we won't go there right now.
How have you been, Dickson? Are you fine?
I've been great.
We are having some winter here in the Northeast.
We're having a winter.
For those of you who didn't know, we are located in New York City.
That's right.
At the
On the
northwestern
banks of the Hudson River.
part of the city, right near the George Washington Bridge, which we can see out the window, right?
Exactly. Which everyone in the country now knows the location of that bridge because of the Bridgegate scandal going on in the state of New Jersey with Chris Christie as our governor.
New Jersey was recently in the news because it hosted the
Super Bowl. I
was working on lectures.
Were you now? Well,
Yeah.
I was sitting at home trying to get into the game, but the first play of the game sort of turned me off when the center for the Denver Broncos put the ball over the head of Peyton Manning. And the tone of the game was set, and it never stopped getting worse for the Broncos.
That guy's going to feel bad the rest of his life.
No, he won't. Nah, he'll. You know what? He has another year left on his contract.
No, the center who hiked him.
No, it wasn't just his fault. The whole team just collapsed. That's what I think. I mean, it's just a matter of, are you ready for this moment in your life? And the answer was Seattle was really ready, and the Broncos were not quite as ready.
Is there a podcast called This Week in Football? TWiP? That's
Right.
it. I don't know anything, so I can't do it with you.
Well, I got my PhD at Notre Dame, and you can't do that without learning.
You're right.
Learning football.
Did you go to games?
I went to all of them. They were fantastic games.
Who's the coach?
Ara Parsegian.
Oh, it's famous, yeah.
He was very famous. That was his first year. His first year at Notre Dame was in 1964. And they went 9-1. And guess who they lost to, Vincent? They lost to the University of Southern California.
I was going to say USC.
In the last 17 seconds
Was that a bowl
of the
game? Bowl game? Oh, that one game that they lost.
That was the game.
All right, let's
change
parasites.
the subject.
Let's do parasites.
Right.
I don't mind talking about the weather. We've
That's
had
fine.
a lot of snow this winter.
Weather looks great right now as we speak. There's almost no wind. I can see the towering clouds of steam coming out of the power plants in New Jersey. So I know that at least we're generating electricity in some regular fashion through PSE&G, which stands for the Public Service.
You know what the temperature is, Dickson?
Electric gas. I would guess 29.
It's -1 Celsius. So it's about 31, 30.
Pretty close.
And it's sunny out, but
It is sunny, but not for long.
We had a
Not for
snowstorm
long.
yesterday,
We did.
and
We did.
we're supposed to get another tonight,
Yes, we are.
starting at 6 p.m. and going till 2 p.m. tomorrow, which will make tomorrow's commute
Horrendous.
miserable.
Which
So-
means that you can work out of your house.
Can't. I have to lecture medical students tomorrow at 11.
Right.
Plus, I have a 9 a.m.
What is your lecture on?
Tomorrow is Introduction to Viral Pathogenesis.
Oh, that's a biggie. You can't stay home for that one.
I'll make it in. It's just that it's going to take hours and hours
Right.
and hours.
No,
and
I hear
hours
you.
and hours.
I hear you.
Well,
Hey.
let's do something interesting. Let's
Sure.
do paper.
Okay, fine.
You picked this paper,
I did.
which was published in Science on 13th of December, 2013.
Right.
12/13/13. How about that?
That's pretty interesting.
And it's called Genetic and Molecular Basis of Drug Resistance and Species-Specific Drug Action in Schistosome Parasites.
Right.
With many authors. You know any
Many.
of these authors?
I know one of them very well. His name is Philip T. Loverde. And he used to be, although he isn't now, he used to be at SUNY Upstate in Syracuse.
Green. Lo Verde means the green, right?
See that? He's an Italian soulmate.
I don't know if it's Italian.
So I knew him very well because he and I were associated for many years at various meetings because we're both worm people. And he apparently adapted very well to the genomics era and became an expert on the genome of schistosomes. So that's what he later on became famous for.
Schistosomes are the ones that look weird.
These are worms that live as pairs of worms, mated forever.
Blood flukes.
Yeah, that's exactly right, Vince.
Shrimp?
Exactly right. No, not shrimp.
Snails.
Snails are the intermediate hosts.
We did a TWiP on them.
We did, of course we did. We did several papers on schistosomes. Interestingly enough, schistosomes were introduced to the quotes-unquote New World during the heyday of enslavement of West African natives to that area. They were brought to both North and South America to work in the rum fields. And Brazil, just recently, they revived this topic, by the way. Brazil, once they become independent of Portugal, because they were part of Portugal for a while, right? That's why they speak Portuguese in Brazil. When Brazil became independent, they offered these people who were at that point enslaved to become free. And that's exactly what happened. But you could guess that because they were from Africa to begin with, they may have brought something with them that the Brazilians probably didn't want in the form of infections.
Schistosomes, among other things.
Schistosomes, among other things, too. Hookworm, schistosomes, some filarial parasites.
Is there still schistosome in Brazil? Because I know it's in Africa.
There's a lot of it. And then you could ask the question, well, this has a snail intermediate host. How does that work? Because if they had brought African trypanosomiasis with them, which they might have brought, it would have never had a chance in Brazil. And the question is then why? And the answer would be because there are no tsetse flies that live in Brazil. Right? And similarly, if they had brought Chagas disease to Africa, there are no tritomids that live in Africa, so therefore there would be no chance for infection in Africa with American trypanosomiasis. So
tritomids?
Tritomids, that's right.
I haven't heard that before.
Really? Reduvids.
Oh, reduvid. I got that.
So the point is that a lot of these infections are localized to places that are specific for their intermediate hosts.
Yeah, of course.
Okay? But one thing that happened in Brazil that was really disastrous for a while was the fact that they brought Anopheles gambii with them in the boats, okay, in the ships that sailed over here. So they actually introduced probably the most vicious biting vector and the most efficient carrier for malaria from Africa to South America. And I think maybe they might have even brought malaria with them as well. So they had a rough time eliminating Anopheles gambii from South America, but they've done it. Okay, they did that. But the snails, Vincent, were already there. They had snails there which could accept the Miracidia that hatch in the fresh water after the people with schistosomiasis defecate, right, or urinate, because Schistosoma hematobium is another parasite like that. But interestingly enough, Schistosoma hematobium, as far as I know, is not found in Brazil or any other part of South America, but Schistosoma mansonii is.
That's the one that is the topic of this paper.
It is, actually. So wait a minute now.
It says here that there are 67 million infections in Africa and South America.
That's right.
With S. mansonii.
That's right. So you'd say, well, wait a minute. We've got a drug for that, right? We know what that...
Praziquantal?
Exactly right.
So what's the problem? Why do we have to publish this?
Hey, well, exactly right. So wait a minute. So there was this other drug, too, though, that they developed, which wasn't very effective.
Before you say that, can you just revise the life cycle?
I could revise the life cycle. No, I better not. I think I better just repeat the life cycle.
So how do we acquire it?
Well, we actually have to swim in fresh water in order for the Miracidia to reproduce inside the snail and produce a stage called the Cercaria.
Cercaria swims in the water.
Cercaria swims in the water. It migrates against the pull of gravity towards the light.
All right, and then when we swim, it goes in our hair follicles?
It does. Actually, that's exactly what happens.
remember,
And then
'cause we
the
did
tail
talk
drops
about
off. The schistosomula, which is the immature schistosome, burrows down through the hair follicle till it gets to a capillary, burrows into the skin, stays put a little bit, maybe 2 or 3 days. It then undergoes morphological change,
Right.
but not size change, because it crawls back into a capillary and gets transported by the blood to the lungs, of all places. When it gets to the lungs, a miracle is. If you're a schistosome, a miracle happens. What is that miracle? The miracle is that you can now look the immune system right in the face, and the immune system can't look back. You can see them, but they can't see you. It sounds like a stealth parasite, and that's exactly what happens, right? We've reviewed this, but I can just emphasize it now because it's one of the defining features of being a schistosome. You can actually camouflage yourself against the particular host species you're in by simply coating yourself with molecules that are reminiscent or are identical or are actually derived from the host that you're living in. So the immune system does a surveillance screen, looks at this gigantic living thing in its bloodstream, but because it's covered with self molecules, the immune system says, well, it looks kind of weird, but I don't know, it looks like you. Hey, you're asking me to find out who you are. This looks like you, okay? I know it's a little weird. You've got things wriggling around here, but I can't distinguish this from your other self. So I'm going to just walk on by, okay?
Walk on by. That's
walk
a
on
song.
by. We're just going to walk on by, right? So we're not going to name that. This episode won't be called Walk On By. We have another better name for this episode.
Do we have a name?
We're working on it. We're
not there yet?
No, we're not, but we're working on it. We're going to come up with a clever way to work in the number 6 and 6, 66. That's
Why?
what we're going to do. Well, because,
It's episode 66?
That's right.
But it doesn't tell you anything about the topic.
Well, not really.
People sometimes complain they can't figure out what the
They're too
podcast
cryptic.
is about. Cryptic like schistosomes.
Exactly.
Right.
So the other point of the story is that once the parasite once this miracle happens, and the surface of the schistosome can now adopt molecules from the host, it can then travel wherever it wants and do whatever it wants, unencumbered by the fact that it has to now somehow avoid the immune response.
So it goes to the
Goes
lung,
to the liver
cloaks
next.
itself, then goes to the liver.
That's right. The cloaking device is acquired in the lungs, and it employs it throughout the rest of its life,
And
which
then
could last
then
up
it
to
has-
30 years.
Has to mate also, right?
They do. They have to make, because the sexes are separate.
And then that produces eggs, which are spread.
Well, they migrate out of the liver as a pair of worms to one of two places. One is the mesenteric vanuoles, which drain the contents of the small and large intestine. And that's how our food gets to us, by the way, of course, and lots of other things too. And the schistosome sits right at the crossroad for our food. And it gets first dibs on whatever we eat. After we digest it and transport those molecules across into our blood, parasite's there. So give me some of that. Give me some. Why? Because the parasite is producing lots of things, right? Producing eggs. So it needs a lot of energy from the host. And that's where it comes from.
The eggs pass out in the feces.
Yeah, there's one other place it could go. It could go to the Venus Plexus, which is the venous circulation surrounding the bladder. Now, why it would want to go there, that's anybody's guess.
Nobody knows.
Nobody seems to
Dead
know.
end.
Well, no.
Does it come out in the urine?
It does. The eggs that they produce, though, you noticed in both places, there's no direct connection to the lumen of either the bladder or the small intestine. So somehow those eggs have to get out in order to enter the environment. And they do so by the stage inside the egg, this little miracidium, secreting proteases, which allow it to actually dissolve its way through the tissues, which creates huge amounts of what we would call pathological consequences. So this worm is really a vicious, long-term, yucky, and not only that, half of the eggs that it produces in both places never penetrate out of the tissues into the lumens. They get washed back by the blood into the liver. They start to plug up the liver becomes compromised, the presinusoidal capillaries become blocked, the liver starts to develop something called portal hypertension. And at that junction, the blood pressure goes up, the blood starts to shunt around the liver. That's bad news for the host because the eggs can now bypass this filter, so to speak, and it can go to other places like your lungs or like your heart. If it goes to your lungs and starts to fill up the capillaries of the lungs, the precapillaries of the lungs, it creates a pressure back on the heart called cor pulmonale. And that's a pathological consequence of a long-term infection. So this is a nasty thing from start to finish, all right? The other thing it does when it bypasses the liver, what about all those P450s? What about all those detoxifying enzymes that we produce in response to all those bacterial toxins that are produced all the time in the gut? We neutralize those right away. We don't need those things. So we employ a lot of proteins, proteases in this case, that are geared up to detoxify all of those bacterial products so that we can enjoy the fruits of our labor, so to speak. And I mean that literally. You can enjoy a fruit or a vegetable or a piece of meat without the worry that our microbiome is going to actually do us in. Once the bypass to the liver occurs, all these toxins have free access to other organs like your brain.
So it sounds like this can be fatal.
In the long term, it can, of course, because what eventually happens is that you start to develop these varices. Do you know what a varice is?
Is it like varicose vein?
No, it's worse than that. It's like if you're an alcoholic and your liver starts to solidify, basically. And as the result of this, you get a cirrhotic liver that places huge amounts of pressure on the blood supply because the blood vessels do not dilate now when the pressure goes up, right? So as the result of that, your collateral circulation, they call it, which used to be in vogue during your embryologic development, can still be recalled anatomically. So now the blood can force its way into these other vessels, which are remnants of your umbilical cord and your esophageal varices develop. These are fragile, enlarged venules, or veins rather, and the veins are very fragile, okay? The other thing that happens, of course, is your spleen starts to fill up with eggs, and that compromises your ability to clot blood because that's where the platelets are produced, and platelets are also produced in your bone marrow, but the clotting of blood is compromised because your spleen is compromised. The next thing you know, these rupture and you bleed to death.
This is a mess.
It's a horrible thing.
But you're very passionate describing this.
Well, I used to teach this. I used to love teaching this one because then I could show that little clip from Arnold Schwarzenegger's The Predator, because I could make him become the worm. So
the clip from The Predator. Didn't we mention
that?
We
did that already. So they can go back and check that out. But the point is, now we're going to talk about a paper, which, wait a minute, for a while, let's go back in history. So what was the consequence of knowing that schistosomes are dangerous, and they're both in the New World and in Africa also? I hate to call it the New World, but just let's say North and South America. It's not in North America. We don't have white snails, but it is in some parts of Central America and all over South America.
Why not in North America?
No, the wrong snails.
Okay. Is there a reservoir host, by the way?
There are reservoir hosts for these. They're mostly they're primates in some cases. In one case, you can even have things like rats that can serve as reservoir hosts. Do we have primates
in North America?
We don't have any native primates. They were all introduced.
We don't have any chimps.
We don't have any non-human primates. We don't have any non-human.
What are the non-human primates, Dickson?
Oh, my goodness. There's so many of them. There are the monkeys and the apes. Didn't we discuss this when Beatrice Hahn was here?
Chimps and gorillas. Then we have all the old world monkeys,
That's right. That's right.
Is a macaque from rhesus macaque, is that a primate?
Sure is. Okay.
Now, you before I interrupted you, For which I deeply apologize.
No apologies necessary.
You were talking about drugs, and in the old days, what they used.
That's right. So in the 50s and before this, there was something called Fuadin.
Fuadin?
Yeah, Fuadin. Fuadin. It was named after King Fuad.
Really?
Yeah, from Saudi Arabia.
Fuad?
Yeah, that's right. And it was a toxic
used for this
drug.
infection.
It was a heavy metal containing drug. It was an awful drug. In fact, it was more toxic than the infection.
Schistosome. They would treat schistosomiasis.
That's correct. Then there was another procedure called the Goldschmidt procedure. Mm-hmm. This was fantastic. They shunted blood from the small intestine to a filter that could trap the adult worms. Mm-hmm. And then they gave a drug called emitin.
Emitin.
And it teed off those adult worms. It caused them to let go of their hold in the small intestine, thinking, of course, they could find refuge back in the liver.
Man, they got filtered out instead, right?
But they tapped the portal vein, and tapping the portal vein allows you to take the blood before it goes to the liver. And they would trap the adult worms on this filter, physically trap them, and remove the filter, and then return the blood flow to the liver. Voila! Adult worms are gone.
Can't imagine.
Patient is cured. No drugs.
Always cured?
No drugs.
Really?
No drugs. As long as they didn't go back. And, of course, there is an acquired immune response that it does develop against the penetrating stage, the Zacharias stage. So the likelihood is that as you get older and living in endemic areas, you actually do not acquire more and more and more schistosomes.
You just get the one.
You get this big dose when you're a kid, and then you just live with it, or you die with it, of course. Mm-hmm. So those two procedures, you've got a very toxic drug, Fuadin, and you've got this other surgical procedure with another toxic drug. Emitin is no trivial drug to give in this case, okay? So then the drug companies, of course, finally realized, well, I guess we better do something here because, I see, because maybe our United States Army and Marines and Navy and Air Force, maybe they're going to go to places that have schistosomiasis.
Right.
And so we need a drug for these people. And so the Army started to work on this. And because at Walter Reed, they started to do some heavy research in the 1960s. Some drug companies picked up the action, and we had a number of drugs that emerged onto the marketplace. All right? One of them was called praziquantel.
Yeah, it's one we use today,
Miracle
right?
drug, right? Miracle drug. I believe that was synthesized somewhere in the 1980s. That's a guess. That's when I remember first hearing about praziquantel. Pardon me. And the other drug that emerged also was called oxamniquin.
Different drug.
It's a different drug, and it works differently. And oxamniquin works better. Now, believe me.
Mid-1970s was
praziquantel is 70s? Good. And oxamniquin was?
No, I got to type it in here.
Yeah, come on.
How do you spell it?
Oxamniquin.
Yeah, look at it. It comes right up here in Google. Oxamniquin was developed in 1972.
Look at that. So co-parallel evolution of drug development. Isn't that amazing? Now, why would there be two drugs for the same infection? And why would one work better in one place and not in the other place? Remember, the organism arrived in Brazil back in the 1800s during the enslavement of West Africans. Mm-hmm. So the organism, has it had a chance to evolve so that it's a little bit different, perhaps, than its African parent infection, so that it's not as susceptible to praziquantel, but it is more susceptible to a drug called oxamniquin than the parent organisms would be susceptible to that same drug? So the answer was, maybe there has been enough evolution to allow for this difference in those two same species. We'd call it cells, Schistosoma mansonii, but we treat them differently. So apparently oxamniquin is much more effective against the Brazilian form of Schistosoma mansonii than it is against the African form, whereas praziquantel is much more effective in Africa. So they were starting to use this drug profusely when they first got it. Oxamniquin in Brazil and in other places too. And lo and behold, something horrible happened.
Let me guess.
Go ahead, take a wild stab at this. you have read this paper, yes? Yeah, so we know what happened. This happens with every drug, right?
The title of the paper, drug
Happens
resistance.
with every single drug ever invented, no matter what.
Even human drugs?
No matter what. For cancers, for
what about
bacteria.
psychological
that's
diseases?
a little bit different.
You
It's
don't
not
get
an
resistance.
infection. No, because then you don't have an evolutionary process.
Yeah, but you could become less sensitive to the drugs.
You could over time,
but in this case,
but I mean, cancer cells are a little bit like an infection, right? Because they're sort of different from you, and they go out of
no,
control.
they can mutate.
And
they
can
have a very high mutation rate.
They do,
Which is
because
why
of
they're
their
cancer to
because
begin
of their
with. So they can become
cell
resistant
cycle
to,
is out of control.
to
That's
drugs. They won't take them up.
Sure.
And so
So
forth.
the evolutionary process is the evolutionary process, no matter where you look. So we've now got a problem. We've got schistosomiasis still in Brazil, and we don't have any way to deal with it. So what is the basis for this?
So they don't
resistance?
use this drug anymore because there's resistance. Out there in the
That's right.
wild, there's
That's right.
resistance.
That's correct. So remember, there are also some reservoir hosts as well. So you could have primates. We have lots of non-human primates living throughout Brazil, right? So that's also true. So now we've known about this for a long time.
In this paper, they want to say, where are the mutations that lead
Yes.
to resistance, right? And
Yeah,
how
that's
do they
right.
work?
That's right. And so they had to wait
Why?
for the molecular tools to develop before they could ask the right questions, because
The whole genome sequence,
even,
maybe?
yes, even though we knew that there was resistance, we didn't have a way of looking at it until recently.
Here's an interesting point they make. They say resistance is recessive.
Recessive.
Well, let's explain that to everyone.
Go for it.
You don't know what it means?
Well, I have a feeling that I know what it means. If you use Mendelian genetics as a basis for a dominant and recessive, then this is not a capital R; it's a small r.
So these organisms must be diploid,
They are diploid, of
which
course.
means they
are.
have two copies of each gene.
Of course they do.
So if let's say we have a hypothetical gene that controls the sensitivity to this drug.
Right. Let's just for
And
argument's
if
sake.
the mutation is recessive,
Yes.
if you have a mutation in one of the two copies
Right.
of the gene, the other gene will still function.
Right. And you can
And-
still kill off the worm.
Yeah. Now,
So you need two little R's to come together.
but that's if it's recessive,
right.
okay? And
Right.
that's what this is.
That
If
is
it were
what this
dominant,
It would be big trouble.
just one mutation, one chromosomal
That'd be horrible.
mutation would be enough to
That's
make it
correct.
resistant.
Then all the worms would have it. So that means that some people are more susceptible to the therapeutic effects of the drug than others, depending on which worms they have, right?
You also say that when you get drug resistance, you have a 500% reduction in drug sensitivity, which is a lot.
That's a lot. Are you nearing the therapeutic threshold for this drug, the therapeutic index, rather? You know what I mean by that?
Are you nearing it?
Well, 500-fold increase in drug in order to kill the organism.
Yeah.
Does that approach your own level of toxicity for
Well,
the drug?
I don't know what it is for this, but
Plus, it's expensive.
yeah. And finally, they say this drug, we're going to abbreviate it OXA,
Yeah.
is species specific.
Right.
Now, Schistosoma
Right.
mansonii is a species.
It is. It certainly
Species.
is. It certainly is.
Then we have Schistosoma japonicum,
Yes.
and
It's relative, found only in the...
Mekong.
Yes. Wow, Vincent.
There's another one.
I'm extremely impressed.
There's another one.
There is one other one.
I remember
It's
it.
the one that lives in the Venus Plexus. Hematobium.
Hematobium.
That's right. It's the one that the Egyptians wrote about in their hieroglyphs as the onset of male menopause.
119 million cases of S. hematobium,
A
even
lot.
more than
That
mansonii.
is right. And it's also got a nasty side effect, and it's associated with a mesothelioma, which is bad news.
It's a lung cancer.
No, not a mesothelioma. I said that was squamous cell epithelioma. Squamous cell
So
epithelioma.
OXA
Yeah.
kills mansonii, but not hematobium
Exactly.
or japonicum.
Exactly. So there must be some specific drug target there that
Okay,
differs
so let's
greatly.
find the drug target.
Let's do that.
All right. So they say here, the first step, Dickson, the complete life cycle of S. mansonii can be maintained in the laboratory.
Correct.
I know you don't work on S
No.
mansonii, but what would that mean?
Well, that means that you can maintain snails for the intermediate life cycle stages, and you can maintain a host species of mammal, because there are reservoir hosts for this. You can take a rat, for instance, and infect it with Schistosoma mansonii. You can infect mice with Schistosoma mansonii. But these infections don't really imitate the full-blown infection of a human infection. Okay?
Right. But they use the snail to make lots of what they call genetically identical single-sex parasites.
Clonal parasites. That's correct. That is correct.
So
by a single miracidium penetrating the foot of a snail and then isolating that snail and keeping it separate from all the other snails so that no more miracidia can go into the snail, you get a single genetic type of either male or female Schistosoma.
Either one. You can tell which one is which by
Well, you
looking
can do a
at
genetic analysis and find out.
All right, because then they do
crosses.
They do crosses.
This is incredible.
How do you take the two miracidia, male and female, and you put them in a dish together?
No. You take snails, separate, uninfected snails,
Yes.
right? And you take single miracidia of either male or females.
Yeah.
And as soon as the snail becomes infected, you isolate a single snail so that when they shed their Schistosoma, it's the Schistosoma that you're going to use.
Right.
It's either male or female Schistosoma. So you take a female Schistosoma from this one and a male Schistosoma from that one, and you put them together in a rat.
Oh, you use an animal for this, and they mate.
That's incredible, though, Vincent. These are just two parasites in the whole body. How the heck did they find each other?
That's amazing.
And the answer is they find each other delightful.
Hey, that's the title.
It's truly amazing.
They find
When I
each
gave my
other
lectures to
delightful.
the medical students, I said it's like two elephants mating in the tall grass. How do two elephants find each other when they are undergoing mating in the tall grass? And the answer is they find each other delightful.
So here's what they did.
Right.
They have a oxa-sensitive isolate
Right.
and an oxa-resistant.
Fantastic.
And they cross them.
By the way, this same stuff was done with malaria, too.
They
But
cross
it's
them?
even harder to do in malaria because you have to deal with the mosquitoes and stuff. It's very difficult work. You can see these parasites with the naked eye almost. This is cool.
So they cross them, and then they get the first generation of offspring, which in genetics is called what?
The
F1.
The filial generation number 1.
That's just wonderful.
Yes. So now if it's a dominant gene,
And then you can get F1 and cross them again, right? And make F2.
you can. And then what do you get?
F2.
If it's totally Mendelian and a single gene, what would be the ratio, Vince?
I don't know, 4-2-2.
Yeah, sure, I don't know. It's
You want me to draw a Punnett square?
25%, 50%, 25%
25, 50, 25 of what?
of dominant hybrids and recessives.
Okay. That's a single dominant gene segregation.
Or a single
Do you
recessive
think
gene
Gregor
segregation.
Mendel fudged his data?
A little bit. I think that's why they defrocked him.
Why was a priest doing experiments?
Good question. And why don't creationists believe in science?
I don't know. These are beyond this podcast. Sorry, I even went there. All right, so they got F1s.
Yeah.
And then they used those to backcross and
That's
generate
right.
388 F2.
That's a lot.
At each stage,
That's a
they
lot.
get a single miracidia,
That's right.
and they test
That's
their
right.
resistance to the
That's
drug by
right.
just growing them in the presence of the drug, right?
Well, they can start the life cycle. No, then they have to start the life cycle, and then they have to try to treat the animal.
It says they measure resistance by exposing adult parasites to
Adult
oxa
parasites,
and
yeah.
monitoring parasite
Correct.
survival.
Yeah.
Because it kills them, actually.
You can do that on a petri dish.
It kills them.
It does.
Did you hear me?
I heard you.
Oxa kills sensitive
I heard
parasites.
you.
All right.
But it won't kill double recessives,
Here's the story. The F1,
right?
and here's the ratio. 25% of F2 were oxa resistant. 25%.
That's right. That's a single
75% of F1 and 25. Wait a minute, I'm getting this all wrong. The F1s, 75%. No, the F1s are all sensitive. You know why? Because if you start out with
you
start out with a resistant, that's got to be homozygous for
That's
the
right,
mutation.
that's right. So
and then there are no
that's how they found
homozygotes
out.
in the F1.
That's how they found out.
Dickson, there are no homozygotes in the F1.
I heard you.
Right?
Yep.
So they're all going to be sensitive.
Correct.
However, when you
cross
the F1s amongst
then
each
they
other,
will segregate according
then
to
you will
Mendelian
get
genetics.
then you're going to get some
That's right.
homozygotes
Yep.
Therefore, and so the
And which way did
percentage
it
is
go? Did it go for
25%.
the
25% are
Exactly.
resistant in the F2.
Well, that's a typical single gene mutation, isn't it?
Right.
Cool.
Okay, so now you have these F1s and F2s,
Right.
and they start to ask, where are the mutations?
Exactly.
Which is not easy, right?
No,
How big
no.
is the genome?
That's pretty big.
All right, pretty big is not a podcast number.
Oh, you want me to actually
Size
look up that
of-
number? They list it here. I believe they do say it, but they give a reference to it. That's all.
Well, I'm
They
just going to
give a
Google it.
reference.
You can do your own thing, and
All right.
I'm
Fine. I
going
thought I did
to,
read this here
find it
someplace.
very
How
quickly.
many base pairs this thing has?
363 megabytes.
That's
Not
big.
megabytes, megabases.
Megabytes.
That's
how
not
big
very
your
big.
computer is. That's not how big your
It's not
genome
huge,
is.
but 363 megabases. They have shown that it's recessive,
Right.
and they have
Because all the F1s were sensitive.
F1 and F2. So
Could
then,
that have happened with F1 dominant?
if the gene were dominant,
Yeah.
well, in the F1 you'd get resistance then.
Of course.
You
So
have heterozygotes.
F1 doesn't tell you anything about recessive or dominant, does it?
It's the F2. Because
Right.
in the F1,
The
the,
back cross.
absence
It's
of
the back cross.
resistance could just be a statistical thing.
Exactly.
So now we have F1 and F2, and they have the parental sensitive.
Right.
They want to try and narrow down
Right.
where this mutation is, right?
Right.
So
Right.
the genome size is about 385 megabases, 385 million bases.
All right.
And you had asked before the size of the human genome. It's 3 billion bases.
Okay, so it's 10 times bigger.
This is much smaller, yes.
Right.
But still, it's a lot of bases.
But you know what? I bet you that if you break it down into genes, though,
So let's say you just sequence the genome of each of these resistant mutants.
sure.
Would that help you?
A lot. Well,
no, because there are too many changes.
You know what might happen? Well, okay, go ahead.
There's just too many. There would be too many differences,
Okay.
and you would know which ones were
Okay.
the important ones. So what they do here is they first narrow it down. So throughout the chromosomes of any organism, there are what are called markers that are always
Right.
there.
Right.
And you can ask, are these markers, which you can detect by sequencing or some other molecular biology technique, do they always segregate with the phenotype that you're looking for?
Yes.
Like in this case, drug resistance. And so they did that. It takes a while
I'll say.
because you have to do the analysis.
Yep.
They narrowed it down. They narrowed resistance down to a single locus near the end of chromosome 6,
Wow.
which is still a lot. But then they can go say, let's narrow it down more. Then they start to use SNPs,
Right.
single nucleotide polymorphisms.
Right.
These are changes known, again, to be in certain places. And they ask, which SNPs go along with drug resistance? So they can get closer and closer to the gene.
Yeah.
And then finally they narrow it down to, about a million base pair region. And they can sequence it and say, what's in here? There are going to be a lot of genes in there, but then they can start narrowing it down even further. Are you okay so far?
I understood all of it. I just didn't get the exact procedures
That's fine.
as to how you would go from the next step to the next step.
Now, first of all, we have to provide a little background here. There is an enzyme that is known. So this drug OXA is actually a prodrug that
Right.
gets into the
Meaning?
worm. It's inactive, and it has to be converted to an active form
Okay.
by the host.
Okay. Do we know other drugs like that?
Tons.
Tons of them.
Acyclovir,
There
which
you
is
go.
a anti-herpes drug, is
Right.
a prodrug.
Right.
Has to be activated.
Right.
So the drug is not active.
Metronidazole.
Yeah.
That's one of those
It's not
drugs, too.
active. It has to be enzymatically
Sure.
converted to an active form in the parasite.
Exactly.
And the resistant parasites don't do that, whereas the
Right.
sensitive parasites
Right.
do.
That's right.
There is an enzyme known to do this in extracts of worms. Okay?
Cool.
It's called a sulfur transferase.
Sulfur transferase.
What is a sulfur transferase?
Well, a transferase in general is some enzyme that moves something from one place to another place.
Right. So this one moves sulfur from one thing to
Something containing sulfur. It
Something
could be
else.
an amino acid containing sulfur. It could be another compound.
right. So they haven't proven it, but the implication is
Right.
that sulfur may be put on the drug to activate it.
Right.
So they started looking at all the genes in this region of this million base pair region of the chromosome,
Right.
And they say they use 3 approaches to prioritize the genes. First, they said that the gene would have fixed non-synonymous differences between the parents. So in the resistant mutant versus the parent, a non-synonymous mutation is one that has amino acid that causes amino acid changes. So first of all, there would be amino acid changes, obviously, right?
One would think.
Between the parent that's sensitive and the
Sure.
resistant one. I'd say that's fun.
Sure.
So 7 of 16 genes in this million base pair region fulfilled this criteria,
Okay.
including 1 of 3 genes showing homology to sulfotransferases.
Bingo.
How is that cool?
Modern biology is just incredible, Vince, and I agree. It is cool. I think that was one of the beauties of this paper, is that there's no level of looking that's impossible now. You can get it right down to the active site of the molecule that's the target for the drug and find out why the drug doesn't work anymore. That's what this paper addresses.
Secondly, they looked at RNA levels to each of these genes.
Yep.
And so they got 16 genes within this million base pair region. 16, right? A couple of them had homology to sulfur transferases. Then they looked at RNA expression. And finally, they looked at the size of the predicted protein. And so they had 4 of 16 that had predicted sizes of 25 to 35,000 Daltons. And this sulfur transferase that had been partially purified previously was about 30 kilodaltons. So
it's within range. Sounds good so far.
So that gives them 6 candidate genes that fulfill all of these 3 criteria that we've just mentioned. They then produced each one as a recombinant protein.
Very cool, very cool.
And then they asked, does this activate OXA?
Nice.
And this, they find that it did. Plus...
That what did? Only one
so
of those
one of
6.
them, sorry.
Only one of those 6 did. That's right.
SMP089320.
Right. Just rolls off your tongue.
From one of the parents. Okay, activated the OXA in worm extracts.
Right.
And then they make RNA-I, interfering RNA, short interfering RNA against this SMP089320.
What would that do then in a worm?
That knocks down the amount of RNA.
Right.
And
it prevents the worm from resisting the drug.
It prevents the worm from activating the drug. So the drug does not kill the worm where you've knocked down the gene.
In a worm that's not resistant. Right. You're making it resistant essentially by preventing it from being able to affect the activation of the drug.
That's right. And in fact, that's what they saw. So when they knocked down this particular gene, the worms, they started with OXA-sensitive worms.
Right.
And they knocked down this gene with RNAi. They become resistant. Because there's no... This gene, remember, its gene product is needed to activate the drug.
That's right.
So if you take it away, you can't activate, so the drug can't kill you.
Exactly.
So biochemically and genetically, they show
it's like proof of concept.
This gene seems to be the one.
It does, and they produce a lot of data that points to that. But then,
now what did you do, Dickson? You have a gene, this SNP089320. You think this is the gene that's involved in resistance?
You do, yes, you do.
What would you do next?
I would look for it in organisms that are related, but not
yet.
Okay. That's what I would do.
What they did.
Yeah, they would express it.
We just did that.
Okay. Then
they
the next
compared the sequence in resistant and sensitive worms.
Sure.
Right?
Sure. Of course.
Now you can, of course, they have
they already had
the sensitive worm. That's the wild type genome.
Yeah, that's exactly right.
And they got some mutants, and they find 2 changes that distinguish the resistant and sensitive worms in this gene. One is a substitution of a leucine for tryptophan at position 256.
Right.
And another is a deletion of glutamic acid at 142. They tested each of these mutations independently to see if they confer resistance. So you take the sensitive protein and you introduce each change separately and you ask what happens. The deletion is the cause of the resistance.
The deletion is the cause. So leucine for tryptophan doesn't work.
It still
activates the drug.
That's right.
But the deletion of glutamic acid results in an inability to alter the drug to make it active.
Right, and hence the worm is resistant.
Got it.
It's a little convoluted, but...
Okay.
So now this is a field isolate. This resistant strain came from the field from people who were treated with drug and drug failed, right?
That's right.
They wanted to say, if we make resistant worms in the lab, will they hit the same gene?
Right.
So they can do that. They grow worms in the presence of the drug, select for resistant ones. Now they can just sequence this gene because they know which gene.
Right.
And in fact, there are mutations in that gene that confer resistance. So that's the right target, right?
Very cool. Very cool.
The way this works, it's a sulfur transferase. It transfers sulfate from a donor called PAPS.
Okay. Like Pep's Blue Ribbon beer?
PAPS, 3'-phosphoadenosine 5'-phosphosulfate.
Okay.
It transfers a sulfur from that molecule to oxay, OXA.
Okay.
And when you put a sulfur on the drug, it activates it, and then it can kill the worm.
And it says how that happens.
We don't know how it kills the worm,
We actually don't know that.
but we know how this activates it.
Right.
Okay?
Right.
And so they took the recombinant protein that they made from their gene, and they confirmed that it transfers sulfate from a donor to OXA. They show this biochemically in vitro. You with me?
I am.
You fell asleep there for a minute.
I did not. No, I was thinking.
Okay.
I just looked like I was asleep.
Now they solved the crystal structure of the protein.
That's amazing.
That's huge.
That
would be a paper in itself.
Tell me about it. They went from A to Z.
But there may be listeners who don't know what it means to solve the crystal structure of the protein, right?
So let's talk about that a minute.
When you solve the crystal structure, you make crystals of a protein.
Right.
Right?
In this case, you have the gene already, so it's easy to express in
Make
bacteria.
the protein, you make a lot of protein.
A lot of protein.
And then you try to find conditions where it makes crystals.
That's the tough part.
And then you bombard them with X-rays.
You do.
And
you do a Rosalind Franklin on it.
The X-rays bounce off of each atom, and you collect
or Wilkins, I should say.
What's called the diffraction pattern, which you can then do magic on and interpret and say, this is what the protein looks
transformation.
like. You do Fourier transformations. And you can determine the exact way the protein looks in three-dimensional space.
I love it. You
can do that with viruses
now. When Daniel Koshland was the editor-in-chief for Science, there were a lot more three-dimensional pictures published because he was a three-dimensional protein structure person. And I loved getting them because if I crossed my eyes just right, I could, without glasses, actually envision the three-dimensional molecule. And I don't see that here. I don't know why they don't publish more 3D pictures for us guys and gals that like to watch 3D movies.
Not only did they solve the structure of this sulfur transferase,
yes,
but they did it together with the sulfur donor, PAP, and OXA bound to it.
They got the whole thing.
So they can see how the sulfur is transferred.
They got the whole thing.
So what they see is that OXA binds in a cavity in the enzyme.
Right.
And the hydroxyl group, which is where the hydroxyl group on OXA, which is where the sulfur is going to be attached, is positioned right nearby.
Cool. And sulfur has an interesting characteristic, doesn't it?
What's that?
It has multiple valencies. It's got a valency of how much?
5?
I don't know anything with 5. The biggest one I know is aluminum. It's got 3. Pardon me. But sulfur by itself can exist as a metal, right? And then you can have sulfur dioxide, and then you can have sulfate, which is this form of it. That's SO4, right? So if the valence of sulfur is 4...
6. It has 6 valence electrons.
6 valence electrons.
That means electrons in the outer shell, right?
And it has some very unusual properties, right? In fact, there are 2 amino acids that we must ingest in order to live that contain sulfur, right? It's methionine and cysteine.
That's right.
So sulfur appears to be a dominant force in nature. In fact, this is a totally different subject, but I will just tell you right off the top of my head that you can't form clouds without sulfur.
Yeah, we
might have alluded to that some other time.
it was in your lecture.
Yeah, that's right.
Yesterday.
That's right.
So basically, on this enzyme,
sulfur is big.
this sulfur transferase is an active site, and right there, the OXA and the sulfur donor are positioned so that the sulfur can be hooked up to the OXA
So I have a
by the
question
enzyme.
here. What would happen if we
Wait
gave...
a minute. Wait.
Go ahead.
Then you can see how the amino acid changed the deletion of glutamine,
Right.
glutamic acid. Sorry,
Glutamic
it perturbs
acid.
the structure enough so that doesn't
It
work
doesn't
anymore.
work. It doesn't work. You
Isn't
don't
that
get
cool?
you don't get an organic addition.
One amino acid.
That's right.
So it makes the protein not able to transfer sulfur to the drug, so the drug's not activated, so it will not kill.
Correct.
So effectively, the worms are resistant by a single amino acid change.
That's amazing.
Wow.
That's amazing.
I think this biology is amazing.
It's lovely stuff, isn't it? What would happen,
Beautiful work.
if you gave a patient activated drug instead? What if you gave it to them in the activated form? Is it toxic for humans?
I'll bet it's toxic. And
I
I'll
don't
bet
know that.
it's poorly bioavailable and has bad pharmacokinetics. That's the last paper we read, all those terms.
Right. Well, yeah.
I think the PKPD is bad and has poor bioavailability.
Got it. I filed that one right away.
Bioavailability
I know it is.
means it doesn't get to where you want it to be
Right.
very effectively.
Right.
Pharmacokinetics and pharmacodynamics are how long it lasts.
Right.
After you give it.
Yeah.
It
Does it
lasts
degrade
10
rapidly?
seconds
Does
or
it
it lasts
say-
a few days or
Like
whatever.
ivermectin stays around a long
I would bet with the
time.
sulfur on, it's not very.
It's maybe not bioavailable.
It may be toxic too.
Yeah. Yeah, because we might have systems that this drug attacks as well.
You see, Dickson.
Because we don't know how it
here
affects
it
the
is.
schistosomes.
So the cool thing about this drug is if it's inactive until it gets in the parasite.
I got it. It's like a time bomb.
So it won't be toxic for us.
No, I hear what you're saying.
Whereas if you give it sulfated already, it might be toxic for us.
That may be what they discovered in their animal studies.
All right, now the last question they address. Do you want to hear it?
I sure do.
They want to know why.
I have read this paper, by the way, but
What's the last question?
Well, I could read this right now.
Tell me.
because
All right, why is it species specific, OXA?
You know, that's what they want to know. They
So
wanna
how did they
know-
do that, Dickson?
They could do a survey of the phylogenetic tree
Of
trematodes.
what gene?
Oh, the sulfur, the
go-
sulfur transferase.
Exactly.
Yeah,
You get an A.
Thank you.
Yeah, and what did they find?
Well, they found out that it's restricted to a very small, narrow group of schistosomes.
It's restricted.
Yeah,
So there are similar genes, but they're very, very different.
they are. But I've got that chart right in front of me, and as you look at the relationships, they start with Clonorchus. Do you know what Clonorchus is?
Yeah, we
Vincent?
talked about that.
That's right.
Is that a trematode?
It is. It's a Clonorchus sinensis. It's transmitted from person to person by eating raw or undercooked fish. Freshwater fish in this case. And so it's got a distant gene that's related to this. And then you see all the family relationships that they've identified here. And there's only one little group of schistosomes out of all the other schistosomes that they can get samples from, both from Africa and from South America, that actually fall into this category of even remotely related to the sulfur transferase that they've been working on from the one from Brazil. So
This, in particular, the Japonicum and Hematobium sulfotransferases have an amino acid change that they say would probably impact OXA binding. So they think that's why it would be not affected by the drug Japonicum and Hematobium.
Yep.
I think they should have done the experiment. Of course, they've already done a lot here.
those are difficult to do, though, because these other parasites are they're more difficult to maintain in the laboratory than Schistosoma mansonii.
Well, you could ask whether the enzyme from them activates the drug so it would inhibit S. mansonii.
Yeah, that's right.
Clever, isn't it?
That would be good. That would be very good.
Or you could probably, they have an in vitro assay for transfer of sulfur.
Yeah.
You can ask,
You know,
do these proteins catalyze the transfer of sulfur
I
to
have
the
a more
drug? It's
basic
easy.
biological question to ask, too. And I wonder if they've ever tried this. And I presume they have, but I don't know the answer. Is they take Siccaria from Schistosoma mansonii female
Right.
and a Siccaria from Schistosoma hematobium male, and they infect an animal with these two Cercaria, will they mate and carry out a viable infection? Will they cross-hybridize?
I don't know. What's the
I
answer?
don't have a clue.
Can you
I'm gonna say
cross
no.
a donkey and a tiger?
Not a tiger, but you can cross a donkey and a horse.
You get a mule,
You do.
but it's sterile.
And you get a Jenny. You can get a Jenny, too.
What's a Jenny?
I think that's one of the back crosses from the F1 hybrid that you get from a horse and a mule. Although I'm sure that someone out there that's listening right now knows that answer.
Of course.
But we could find out just by typing
Well,
it out.
I'd like
of
to hear it
course.
from our listeners.
I would, too, because we need to hear from listeners.
We only have 2 email today.
We've just got 2
It
paltry.
makes me feel lonely. Like we're talking into the
void.
Anyway,
Or
so
they're
Dickson, let me
avoiding
ask
us.
you a question.
Please do.
So this is a beautiful study where they've shown
It is.
the basis for
It is gorgeous.
OXA resistance in S. mansonii.
Right.
They've crystallized the protein. The
Yep.
sulfur transferase shows how it works, shows how the mutations
That's right.
affect its
And
activity.
it's a phylogenetic analysis.
We don't even use this drug anymore, Dixon.
We don't.
Why do we
no.
care about any of this?
Because we're interested in the biology of this.
I totally agree. I just want to know how you sell this.
To whom? Or to WHO?
I have a bridge. Are you interested to sell? I have a
A
bridge.
bridge?
Yeah.
You have a bridge?
Yeah.
Is this one that replaces all my missing
it's a
teeth?
bridge, Brooklyn
or is this
Bridge.
one you cross? I think Christy is trying to sell one also.
Yeah, he would like to.
Yeah, I'm sure he would like to. He's trying to sell the idea that he wasn't involved, I think. We don't know
So
the answer
you now
to that,
have a
by the way.
structure of this sulfur transferase.
Yes.
Maybe you could use it to design inhibitors so it won't be activating
And why don't we use this drug anymore?
because
The resistance level has gone up.
so maybe
right?
we could design an inhibitor of the sulfur transferase.
Ah.
No, that wouldn't work because the resistance
It's a
doesn't.
deletion mutant.
The resistant parasites do not activate the OXA.
They don't. That's right. They resist activation. And it's a deletion mutant, too, so you can't really work around the fact that it's missing something rather than has altered something. So leucine for tryptophan didn't work.
What they're saying here, though, right, is that
See, I remember
they could
this
use
paper.
the structure of OXA bound to the sulfur transferase to try and design other OXA to redesign it basically so it's a better.
But meanwhile, we've got praziquantel, which works just as.
Do you get resistance to praziquantel?
The claim is yes, there is some R1 resistance. There are 3 levels of resistance, as we all know, with regards to most drugs, and we're at the R1 level for praziquantel in Egypt.
What is the mechanism of action of praziquantel?
Praziquantel is an inhibitor of calcium flux. As I remember. That's just off the top of my head. I think that's how it works. I think it blocks a calcium channel.
Well, here it is. The mode is not exactly known, but there's evidence that it increases the permeability of membranes in the schistosomes towards calcium ions. Very good, Dickson.
Thank you,
You
Vincent.
got it.
Yeah, I used to teach this, remember? Some things
So it
you
makes
don't
the parasites contract,
Yeah.
resulting in paralysis.
That's right. And it's also thought, by the way, that praziquantel can't work in a new infection. It has to work in an older infection in which antibodies have been slightly successful in damaging the tegument to allow praziquantel full access to the membranes.
Do you get resistance?
You do.
And where is the target?
We don't know.
Why not?
We don't know. I don't know why we don't know, but we don't.
I'm looking it up right here.
Okay.
2012.
That's pretty recent.
Resistance to praziquantel.
Right. Is that a paper produced by
Parasitology research. It doesn't mention.
In the abstract, it doesn't say how it's resistant.
No, it doesn't say.
No.
There are quite a few papers about resistance.
Yeah, no, I remember that everyone said, they can never become resistant because they don't reproduce that rapidly because it doesn't raise that many mutants, but that's not true.
1994 paper says it's the
yeah,
first demonstration of resistance.
That's right.
to
That's
praziquantel.
exactly right. You know why? They used to have an ad campaign for the use of praziquantel in Egypt on television, and it showed 2 Egyptians in the rural area of Egypt, farmers, and one would say, "What? how could you possibly catch schistosomiasis? And the other one would say, well, because I have to wade in the water. He says, well, in that case, you better take your praziquantel because that's the drug of choice now. And so every time that ad played, there would be a run on the market. And it's an across-the-counter drug, unfortunately, and it was not by prescription. People would just run out and buy it and just give it to their kids, hoping to protect them against the infection. And, of course, a lot of times they did, but in other cases, of course, as you know what happened, they selected for low-dose resistance. And I'm not sure if it's up to high-dose resistance yet.
Would you like to read the 2 emails?
Yeah, I'd love to. I would love to. I'd love to.
We should tell people that our listeners should send us email. Well, Dickson,
I know.
If they don't want to talk, it's fine. We can talk.
I know what they're going to say to us, though. If you don't talk, we don't talk.
All right, the first one is from CN, who writes, "Greetings, professors."
Yes.
Are you with me?
I'm not only with you, I'm reading along with you.
After having listened to your discussions on Plasmodium TWiP 64,
yes,
I explored papers on treatment options that are actually available. After having read some papers, I realized that one of the main roadblocks are the Hypnozoites.
Uh-huh.
If I have got it right, apart from the fact that it is involved in reactivation of infection, its dormant nature allows it to evade the antibiotics. My first thought was, can the Hypnozoites activate the immune system, and can it be a vaccine target? Your thoughts on this would be very helpful.
Well, I mean, this is a guess. I'm not basing this on anything I've read or heard, but the word hypnozoite means just that. It's like looking at a spore of a bacteria or of the pseudocyst for Toxoplasma gondii. If you look at those structures for metabolic activity, you'd be hard-pressed to find any. So drugs don't act on things unless they're active, unless they're doing something, unless they're exhibiting metabolism. And so if hypnozoites are not metabolizing, if they're dormant, then drugs are not going to be very effective against that. Just like you can't defeat spores in the ground for anthrax or for Clostridium, for instance, and you certainly can't knock out the pseudocysts of Toxoplasma in the brain tissue of an animal because they're not metabolizing at that point.
Could you use the Hypnozoite to immunize someone?
They're very difficult to come by. I mean, there are not that many of them. How would you get a lot of them
You'd have
together?
to find an antigen and express it.
Well, that raises the point of what antigen would you select for any of the stages of malaria?
Well, you'd have to first show that they could be protective.
That's true. You'd have to do that.
And that's very difficult, is what you're saying.
I'm very much saying that. And in fact, the reason why they're so successful is because they're silent. They just sit there and do nothing, and then all of a sudden, for some reason that we don't understand, and it's not related to the waning of an immune response or a up and down level of an interleukin that we're not aware of, these things pop up randomly almost until they run their course, and they can live for up to 5 years in the liver without doing anything. So they're mysterious little strangers, I would say.
All right, CN continues. In connection with the search, I found a paper in Nature, cited below, which I find is a worthwhile discussion topic. On malaria, the paper is called Host Cell Sensors for Plasmodium Activate Innate Immunity Against Liver Stage Infection.
Right. So this is the sporozoite going into the liver. And there are receptor molecules on the sinusoidal tissues, on the sinusoidal epithelial tissues that the sporozoites have to attach to first. And I presume that what we're looking at here is some studies that have taken advantage of knowing what those receptor molecules are.
So basically they find that the liver has sensors for detecting Plasmodium, and that when they sense it in the liver, they make a type 1 interferon anti-parasite response.
Are these DNA molecule sensors or something? We don't know what part of the parasite they're detecting.
Well, the
toll-like receptors. I remember us discussing this in our TWiP. not basic lectures, but we've had lots of TWiVs on toll-like receptors. And
yeah, so often for viruses, what's sensed is often nucleic acid.
Exactly, but in this case, we're not sure.
No, they don't actually identify what the molecular pattern is.
Right.
They just show that it's there. They can tell because downstream of the sensor, they know the pathway.
Sure.
But they don't know what it is, which is kind of interesting that they haven't figured it out. Anyway, so the plasmodium's detected.
Right.
Maybe it's RNA, plasmodium RNA.
Right.
And then that induces interferon, which is protective.
Right.
They have a figure here showing in the liver cell the activation of this response, and they have plasmodium RNA question mark.
Right.
So,
want me to read the next one?
No, I don't. I'm not done with this one yet.
Oh, I'm sorry. So sorry.
Thank you for all the time spent on educating us, Twix fan. All right, you can read the next
That
one.
was very nice of them to say that. Kim writes.
Speak into the microphone.
Yeah, I'm trying to read at the same time.
It's a gift, you know.
Yeah, it's a gift.
develop it.
That's right. I'm abusing the gift right now. How's this? Is that better? Kim writes, "Hi, Vincent and Dickson. I just want to let you know that I would very much like to attend a Coursera course on ecology led by Dickson. I really enjoy episodes such as the TWiP 65, where ecology, parasitism, and various infectious diseases are intertwined, and would love to learn more about the ecological aspects. However, as I study purely infection biology, I don't have the mental strength to start studying ecology on my free time, as I feel it is hard to know from where to start. Here, I think a web-based course would be a great way for me to learn more and would keep my interests up, with Dickson spicing up the potential future lectures with references to both parasitism and vertical farming. I really hope this materializes." Well, that's a great suggestion, Kim. I've been on the brink of developing an online course for ecology because I've been teaching this to various levels of undergraduates and graduate students for the last 15 years. I really enjoy the subject, and I really enjoy the application of the subject to infectious diseases and to other things such as agriculture. And as an aside, of course, today in the United States, Kim is from Uppsala, Sweden. In the United States today, the United States Congress, both the Senate and the House of Representatives, are about to agree on a farm bill which approaches $1 trillion.
What is a farm bill?
Exactly.
Give subsidies? Is that the idea?
It's got everything in it. It's got food stamp money. It's got farm insurance money. It's got money for innovation, for outdoor farming, but I don't think it has any money whatsoever for urban agricultural initiatives. I'm not sure about it because I haven't seen it in its full form. It's a huge document, and it's one which the Congress passes over, much like the Pope blesses the audience at Easter. Everybody sits out in the audience, and the Pope raises his hand and makes the sign of the cross. When the Farm Bill comes up for vote, it's like there isn't a single Republican or Democrat that would dare vote against it, because it's really heartland America that we're trying to support here.
Did you say $1 trillion?
It's almost $1 trillion.
Wow. It's like the defense budget.
It's no, not quite. Nothing is like the defense budget.
How much is defense?
This is for only 5 years. This is for the next 5 years. By the way.
Defense is $1 trillion for 1 year.
Exactly. It's huge. I mean, you could look it up right now too. The numbers are out there.
That's
But
okay.
And by the way, speaking of that,
yeah.
At the end of this month, unless we do something, we can't raise the debt ceiling, so we can't borrow money to pay off all these programs that we've got going.
Yeah, what happens?
And again, we've got the Democrats.
I thought they passed the budget.
Nah, they did. They passed it right up to the end of February. But we're at the same position we were 3 months ago now. It's crazy.
This course, you're going to charge people to take?
No, I'm not going to charge. I don't know what I'm going to do.
Why don't you do it on Coursera?
I don't know what I'm going to do.
I can show you. I
would like to know what to do, though. I would love to take lessons from you.
I will let you record them right here in the TWiP studio.
It would be great.
Well, I'll help you split them up, and then we can get Columbia to help you put them on Coursera. I think you would.
Let's try.
Why don't you do that? You won't make money, but you will enrich the world.
Why don't I do it?
No, he's looking at me saying, I want to make money. I want to be rich.
I'm too old to want to be rich. I am rich in experience and in life in general.
By the way, do you know what the current temperature is when Kim wrote her letter in Uppsala,
well,
Sweden?
it must have been very cold.
-7 Celsius.
Right, that's cold. Now
That is really cold.
Friends, that's cold.
Have you been to Uppsala?
I haven't been to Uppsala, but I've been to Stockholm, and Stockholm gets very cold. I've been to Linköping.
Hey, I was invited to Sweden.
Really? When are you going to go?
August.
Nice. That's a great time.
Will it be nice then?
Oh, it'll be lovely. It'll be absolutely lovely.
Do you want to know where I was invited to? I have to do some
Are
TWiPs in there.
Are you going to receive a prize of some sort?
No, I'm not getting a prize. No way.
Can I hold your coat?
No, I don't get these things.
I don't know. Where are you going to go?
I was invited to...
Göttingen?
Hang on. Smogen.
Schmogen.
Smogen, on the Swedish west coast.
Swedish west coast. West coast? The Swedish west coast?
That's what it says.
I don't think so.
It says the
Sweden
Swedish
doesn't
west
have
coast.
Where's the west coast of Sweden?
Dickson, the guy is from Sweden, he wrote.
Sweden goes around the tip. That's just a little past the Narrows between Denmark and Sweden.
Yeah, between Sweden and Finland,
it's
not
near
Denmark.
Oh, down.
West,
It's way.
I'm sorry, west coast, you're right, between Sweden and
Denmark.
Denmark. There's a very little bit there,
Yeah, that's
I
where
guess.
Göttingen is. Göttingen is there,
Gothenburg.
too. Gothenburg is exactly what I meant to say,
But
of course.
in Copenhagen is right there,
Yeah, that's
too.
not far away. You know what they're doing, Vince? They're thinking of putting a bridge across.
It's not very far, is it?
No.
What's the
it's
name of
only
this
23
place?
miles.
So Malmo is there.
Malmo is there. That's
Lund,
true. Malmo is a
Helsingborg.
great city.
What did I call this place? You've
Begins
already
with S.
forgotten.
Begins with
You've
S.
already forgotten.
I did. Well, so did you.
That's because I'm
But I'm not going. You see, I'm not going.
Smogen.
Smogen. It's got an umlaut over the O.
2 dots.
That's an umlaut.
It's a
Schmogen.
It's
Schmogen.
in Götaland County.
Götaland. So I've been to Linköping, which is further north than that. It's between
There it is, Dickson. See,
Gothenburg.
there's the very tip of.
Now I see it now.
And here's
I see it.
Denmark here, right? Right here.
Okay.
So there's Denmark, and here it is. Here.
Keep really
keep zooming out?
Keep zooming out.
Oh,
So where does Norway start?
Wow.
Yeah,
This
exactly.
is pretty. Oh, that's not even.
No,
It's
right.
north of Gothenburg.
Exactly.
Wow.
Exactly. So Norway is over here, I think.
There it is.
And
It's in
there's the
that.
border. The border. You're almost in Norway. You're almost on
I
the
can
border.
fly to Oslo and take a train.
You could do that.
I like Sweden. I
It's
have
a
to
great
do
country.
a TWiP because there are lots of virologists.
You
in
will
Sweden.
also love the food. The food in Sweden is very, very good. It's a lot of fish, a lot
Skål.
of. They do a fair amount of alcoholic ingestion.
So August will be nice and warm, right?
It'd be great. You're gonna have a great time. A lot of smoked
I'll
fish.
go for like a day, right?
Right. At least a day, events. And I'd take 2 days just to be
I
brave.
have to do a couple of TWiPs.
That's good.
All right.
That's great.
That'll do it, Dickson, for TWiP number 66. Why don't you come with me to Sweden? We can do a TWiP.
In August, I could be tempted to do that.
If I had money, I'd pay for you.
No, see, now you know why I want money. Because if I didn't want any money, then I couldn't even think about joining you for this
It would
trip.
be fun. We could do TWiPs and TWiVs.
We could do that, and I could do with my Swedish chef imitation.
How do you do that?
From the Muppets.
How does that go?
You want an invitation right now? You remember the Swedish chef that used
No,
to chase
I
the
never
chicken
watched the
across?
Muppets.
You never watched the Muppets?
No, I don't watch TV generally, Dickson.
That's true, you don't. So you're culturally
I'm not deprived.
Sheltered.
No, I am not sheltered. I used to read instead. I mean, I'm not being snobby, but I really liked reading.
I
still do.
read too. I read, but I also share my time between reading and television and music and fishing and cooking.
Dickson, where can you find TWiP?
You can find TWiP on many, many places. Probably you can find it on iTunes, and you
Yes.
can find it on something TV, which stands for an
TWiP.
island in the
That's
Pacific Ocean.
TWiP.tv. That's
TWiP.tv.
TWiP.
TWiP.
Is on iTunes and microbeworld.org/TWiP. And if you like us, people should go over to iTunes and just rate the show if you haven't done that
we're
already.
losing our listenership here, I think.
No,
we should
don't
do this more
I think
frequently.
we're losing it. It's just that they're becoming quiet. I think they're tired of us
They're quiescent.
talking.
They're
Probably.
quiescent.
Think we should get some guests?
Yeah, I do.
We
I
should
do.
go to a guest house. which would be Gasthaus, right?
We know some people that want to be on the show, and we should invite them. Set it up.
Also, send your questions and comments to TWiP at twiv.tv.
That's right.
Dickson Despommier, thank you for joining me today.
It's, always a pleasure.
Couldn't have done it without
As
you.
Alan Dove used to say,
Always a good time.
Always a good time.
It's very funny.
That's
you can
right.
find Dickson at Trichinello.org. Trichinello is back.
It is.
Huffington Post,
Well, we're working on that.
verticalfarm.com,
Oh, yeah.
and medicalecology.org.
Got it.
All those wonderful sites. Thank you, Dickson.
You're welcome,
I'm
Vincent.
Vincent Racaniello. You can find me at virology.ws. The
Right.
music on TWiP is composed and performed by Ronald Jenkins.
But today we may change that music just to match with the number of the show that we've got here. I suggest strongly that we reconsider.
I can't. It's copyrighted. Not allowed to do it.
Oh, yeah, no. This is...
We use Ronald Jenkins' music with his permission. And you should check him out at ronaldjenkins.com.
I think he's great, by the way. I really do think
You've
he's
never
great.
listened to one episode.
You are so wrong. You are
I
so,
love to bug
so
you.
wrong.
Really? You've listened to them?
Of course. But I get tired of listening to my own voice. Come on,
Yeah, so do other people, apparently.
Apparently so. That's right.
Been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.